Hello everyone, welcome to Clarity for Sure. Today in this video we are going to discuss some of the important topics related to biotechnology. This video is very important for your upcoming UPSC CSE Prelims examination. So without any further delay, let's get into the session. See, before moving into the session, I just wanted to tell you guys that this session will be dealt in two parts, whereas today in this session we will be discussing the concepts related to some of the topics of biotechnology, whereas in the next section a detailed MCQ will be discussed from these topics, which can be immensely helpful for your upcoming UPSC prelims examination. So coming to the first topic of the day, it is about a gene. So we will try to understand the basics of a gene. What is a gene? A gene, it is nothing but a small section of a DNA that contains the instructions to make a specific protein. Why these proteins are important? Because these are the proteins that will control how our body should grow, develop and function. Okay. Now coming to the basic difference between a DNA and RNA, we all know that DNA is the deoxyribonucleic acid and RNA is ribonucleic acid. The sugar component is deoxyribose in DNA and it is ribose in RNA. The strands, we all know that DNA is a double stranded in a helix format, whereas RNA is a single stranded. And the basic difference between a DNA and RNA is the nitrogenous base. Here we have adenine, thymine, cytosine and guanine, whereas in the place of thymine we have uracil in RNA. So it is adenine, uracil, cytosine and guanine. Base pairing, see, both DNA and RNA will be always present in the form of pairs only. It is adenine and thymine and it is cytosine and guanine. In RNA, it is adenine and uracil, it is cytosine and guanine. So what is the location of a DNA? We all know that the location of a DNA will be mostly in the nucleus, whereas the RNA is present in nucleus and the cytoplasm. And what is the function of this DNA? See, DNA is a place where it is a repository that stores the genetic information, whereas the RNA will transfer the genetic code for the synthesis of protein. Okay, it is a repository, whereas RNA acts as a transfer medium of transferring the genetic code for the synthesis of protein. Stability, DNA is more stable because of the double strand and it lacks the OH group, whereas RNA is less stable. Why? Because it is more reactive due to the presence of this OH group. Okay, types, we all know DNA is only one main type, whereas RNA, it has three types: mRNA, tRNA and rRNA. And replication, see, DNA is self-replicating, whereas the RNA has to be synthesized from DNA by a method called transcription. Okay, so this is the basic difference between a DNA and RNA. Having said this, we will try to understand some of the important gene editing techniques, or the techniques that are used in the field of gene editing. Okay, the first comes your recombinant DNA. What is recombinant DNA? See, recombinant DNA is nothing but it will add a useful gene from one organism to another organism. It means it will cut and paste a gene from one organism's DNA to another organism's DNA using a plasmid. Okay, here the cutting and pasting of gene will not be precise, rather it will be done without a precision. So example of a recombinant DNA is making a drought resistant crop, whereas a drought resistant important trait gene is taken from one particular plant and that will be inserted into the gene of the another plant, making the outcome plant as a drought resistant. And what is gene editing? See, gene editing is a precise technique. Unlike recombinant DNA, gene editing is a precise technique where it precisely fixes or changes a specific part of DNA. This is a very important point. And a proteins like the nucleosides like Cas9 or Cas12 will act as a molecular scissors that will edit the a place of DNA where we need to change a specific part of DNA. An example of this is fixing a sickle cell disease mutation. It means that it will cut precisely where a mutation in a gene will occur, so that if that editing is done, that mutation gene will not be replicating. Hence, the sickle cell disease can be fixed. Okay. What is a gene knockout? See, gene knockout is nothing but it will turn off or remove a gene to see what will happen in the absence of a particular gene. See, this is mostly used for study purpose. For example, if you are going to take a crop and a crop is having a certain trait. Now, scientists what we will do, they will use this gene knockout technology to turn off or remove that particular gene from that particular plant. That particular trait will be removed from that particular plant. So what happens if that trait is removed, how the plant or the organism will behave. So that is all about gene knockout. So what is gene silencing? See, gene silencing is nothing but it is opposite to the gene knockout. Whereas in gene knockout we will be permanently removing, or we will be permanently turning off a particular gene. Whereas in gene silencing, what we will do, we will be turning off a particular gene temporarily without changing any structure to the DNA. Whereas in gene knockout, when there is a removal or turning off a gene completely, there will be a disruption that will be done to the DNA. Whereas in gene silencing, that disturbance will not come. Instead, they will use a small RNA molecule that we will call as a template. They will use a small RNA molecule to block that particular gene from making a protein. So for example, we can see that if a virus is being taken and we are going to silence a particular temporarily silence a particular gene, for example, the reproduction gene in that particular virus. So what happens? This RNA molecule will block the gene from making proteins, hence the reproduction will not happen. So this will prevent virus from multiplying in plants, so that the plant will know the disease attackness in the plant will also be reduced. So this is the basic difference between a gene knockout and a gene silencing. What is a protoplast fusion? See, the keyword here is it is a mixing of two different cells to combine their good traits. For example, there are two plants where one plant is drought resistant and one plant is of high yielding capacity. Okay, but the drought resistant plant is not high yielding and the high yielding plant variety is not drought resistant. So we will be taking the two good traits, the drought resistant trait and the high yielding trait. We will be combining both these genes and we will be creating a new new form of plant which will have a traits from both these plants. Okay, some example which I have given here is creating fungus resistant potatoes or seedless grapes. Okay, so this is an example for this protoplast fusion. I hope the technique, some of the important techniques related to gene editing is clear to you now. Now we will see what is the difference between a recombinant DNA and a protoplast fusion. See, the gene source for this recombinant DNA is from other species. It means it is a cross species gene editing technique, whereas in protoplast fusion it is from a same genus, that is from closely related plants or organisms. Method: here we will be inserting a gene using a vector, that is a plasmid, whereas in protoplast fusion what we will do, we will be fusing the entire cells to mix the genome. Example of this recombinant DNA is like GM crops, example the insulin bacteria, and for protoplast fusion it is new plant varieties with the combined traits from a closely related species. Okay, GMO status: yes, recombinant DNA is a genetically modified organisms because we are inserting a new gene from another species, whereas in protoplast sometimes it is considered as GMO, but it is often debated. Okay, understood this concept, we will try to understand the concept of gene regulation. What is gene regulation? See, gene regulation is nothing but it is a control of the timing, location and amount of gene that has to be expressed. For example, it is a method where a gene regulation will be done on how a cell decides when to express the gene, when to express them, or how much protein to make from that each gene. Okay, this is an important concept because we all know that the 2024 Nobel Prize in the field of has been provided to the field of gene regulation. Okay, there are four important steps in this gene regulation. First is the transcriptional step. What is a transcriptional? See, in this step, it is a step where a gene controls, a control will happen whether a gene's DNA is transcribed into RNA. That means it will act as a on or off switch. Okay. Next, after this transcription, what happens? This is a place where a modification of RNA will be taking place. For example, either it is to splicing up of the RNA or to check the RNA stability before it becomes a template for protein. Why RNA becomes a template for protein? We have seen in the previous slide also, where the RNA, a small RNA molecule will act as a template, and that is in turn used in the gene editing technique either to silence a gene or either to edit a gene. Okay, that will act as a medium. This RNA, the modified RNA, will act as a medium for the further steps in gene editing. What is translational? See, translational is nothing but how effectively the mRNA is used to make the protein. mRNA is nothing but the messenger RNA, because we have seen in the difference of DNA and RNA, where we have seen that the role of RNA is to transcript. Okay, so that translate the protein, translate the molecule into the protein. So it controls how efficiently the mRNA is used to make the protein. What is post-translational? Post-translational is nothing but it will modify the protein after it is made. In the translational stage, the protein will be made. In post translation, it will modify it, whether it has to be activated or it has to be deactivated. That is the post translational stage. So these are the four methods, four steps in levels in which a gene regulation takes place. I hope the concept of gene regulation and the levels of this gene regulation is clear to you now. And we will try to understand why this gene regulation matters. Because see, this gene regulation will actually help us cells to specialize, and it will help the cells to form a new tissue or organs from the proteins that has been synthesized in the third step, that is the translational step. And this will also help for the adaptation of quick response to stress, hormones and environmental changes in an organism. And this gene regulation also plays a very good role in tackling some of the disease like cancers, autoimmune disease and genetic disorders, because this gene regulation, when a gene regulation is done to a particular DNA, the diseases that are causing a gene that is going to cause a particular disease can be regulated. It means that it can be minimized. Okay, this is a importance of a gene regulation. Now coming to mRNA, because we have seen this mRNA is an important it is an important RNA throughout this gene regulation process. See, this mRNA, which is nothing but the messenger RNA, is a temporary RNA copy, okay, that is made during the transcription in the nucleus. This mRNA will carries the genetic instructions from the DNA to the ribosome, where it is translated into the specific protein, and this mRNA will that translated protein will act as a crucial link between a gene and the protein expression. So the role of mRNA here is to carry the genetic instructions from the DNA to the ribosome, where in the ribosome it will get translated into a specific protein which acts as a crucial link between a gene and the protein expression. So this is an importance of this mRNA, which is a temporary RNA copy, because we have seen this, the RNA are available in three types: tRNA, mRNA and rRNA. This mRNA plays a major role in this gene regulation. Okay, I hope the gene regulation topic is also clear to you now. And we have been discussing the terms related to transcription, translation, everything. Now we will see what is the basic difference between a translation and a transcription and translation. See, this transcription is a process where it is a process of copying a DNA into mRNA, whereas in translation it will translate that mRNA, because we know that mRNA is a carrier which will carry out a genetic information from DNA. So that genetic information will be decoded from mRNA to build a protein. Based on that genetic information that has been given by DNA, a protein will be built. Okay, what is the location? It is in the nucleus, and the nucleus in the eukaryotes, and it will be present in the cytoplasm in the prokaryotes, and in translation it will be in the cytoplasm, which will be in the ribosome. Okay, template used here, the template for transcription, that is a copy from DNA to mRNA, the template will be DNA, whereas in the translation the mRNA will be a template. Okay, what is the product formed here? It is the mRNA. That is, in the transcription the product is messenger RNA, whereas in translation the product is the protein, that is the chain of amino acids. That chain of amino acids has been made from the genetic information that has been given by the DNA, and that has been carried out from mRNA. Okay, what is the enzyme involved here? Here the enzyme is RNA polymerase, and here the enzyme is the ribosome enzyme. What is the base pairing? We all know that during the transcription the base since it is RNA, the base will be AU or it will be GC, because we know RNA uses uracil in the place of thymine. And what is the translation here? It will be in the form of codons, and it will be matched with anticodons. That is, the transmitter RNA is the anticodons. Okay, codons or mRNA and anticodons are tRNA. What is the purpose? It is to create a RNA copy of the gene. And what is the purpose of translation? To synthesize the protein based on the mRNA instructions. Instructions is given by the DNA molecule, and this mRNA will carry out the DNA instructions, and finally it will be synthesizing the protein based on the mRNA instructions. I hope the topic of gene regulation, the difference between the transcription and translation, is clear to you now. Okay, next is an important current affairs topic from your current affairs. See, the scientist from Indian Institute of Bombay has come up with the first homegrown gene therapy. It is the name of the gene therapy, that is next CAR-19 CAR T cells, which is a therapy for cancer. Okay, what are these CAR T cells? See, CAR T cells are type of immune cells that have been genetically modified to better recognize and kill the cancer cell. So this is the type of immune cell that has been developed by the scientist of IIT Bombay, which has been genetically modified. What is genetically modified? A foreign DNA has been infused into this particular immune cell to act better, to recognize the cancer cells better and kill it. Okay. What is the procedure of this particular CAR T cell therapy? See, scientists will be taking the cells of the patient's own T cells. We all know that T cells are the repository of the WBC, and we also know that C cells are cytotoxic in nature. Okay, now they will take that T cells from the patient's own cells. They will reprogram that cell in a lab with the special receptor. That receptor is known as the CAR. What is a CAR? It is chimeric antigen receptor. Okay, this CAR, that is the chimeric antigen receptor, will target the cancers that is being grown in the body of the patient, and it will it will kill that particular cancer cells. How? These CAR T cells will be grow in large numbers, and when that cells are infused or injected into the cells of the cancer patients, this CAR T cells have the ability to better recognize that cancer cells and kill them. Okay. So as I already told you, this CAR is a synthetic receptor added to the T cells to help them recognize and kill cancer cells by targeting specific proteins on the surface. The proteins on the cancer surface, this CAR cells will be targeting that specific protein, and it will be killing that particular cancer. So here is the visual diagram of this particular CAR T cells. So see, T cells are uh, or collected from the blood of a patient, and based on in this T cells, a synthetically grown CAR cell will be grown. This CAR gene, the gene of this CAR, chimeric antigen receptor, will be inserted into this T cell, and what happens, this T cell in turn will be grown in lab, and these will be grown in millions, and now they will be infused this lab grown CAR T cells into the patient blood again. Now what will these CAR T cells do? This will bind into the cancer cells, attacks the specific protein of those cancer cells, and these cells will kill the cancer cells. So this is one of the methods in which a cancer can be cured. So this is just in the initiation stage. So the outcomes of this method, the pros and cons of this methods are yet to be known. And one con of this particular CAR T cells is that this method cannot be used to target big tumors like breast cancer, etc. Okay, so that is one of the cons of this particular CAR T cells. Since this stage, this CAR T cells still it is in the initiation process, we should wait for the better outcomes from this particular CAR T cell cancer therapy. Okay. Now, DNA repair. See, this is one important topics from your current affairs, that scientists have come out with a DNA repair mechanism where the cancer cells by itself is repairing a disruption that has been made to that DNA. They have found that molecule. So silencing that molecule or targeting that specific molecule in that DNA, we will be able to get rid of the cancer. We will be able to cure the cancer. So this is the background of this particular DNA repair. So now let us see the basics of the DNA repair. See, DNA repair, it is a natural process that helps to fix the damaged DNA to maintain a genetic stability. Okay, this is essential for life, which is occur in both simple and complex organisms. However, cancer cells exploit the systems to survive the treatments. This is what I was telling, that cancer cells, they are repairing their DNA, rupture of DNA that has been done by the treatment. So the cancer cells are not killed, rather they are repairing their own DNA, and they are growing. Okay. Now, what is the enzyme that has been found by the scientists? The enzyme helping cancer resistant treatment is TDP1. This is the enzyme that repairs the DNA damage, especially the kind caused by chemotherapy drugs like, okay, camphotericin. Okay, this cancer cells is using the enzyme called the enzyme called TDPN to repair the damage that has been done to their own cancer cells by the chemotherapy treatment. So what scientists have done? So scientists have researched and found out that if this TDPN enzyme is targeted in the cancer cells, we could able to block the cancer cells ability to repair the DNA, which in turn will increase or enhance the effectiveness of chemotherapy, and it will also reduce the drug resistance, because we have seen the drugs that are being used in chemotherapy is not doing it actual purpose. Why? Because of this TDPN. So targeting this TDPN, either silencing off or either cutting it using the CRISPR-Cas9 or Cas12 technology will be immensely helpful, so that the cancer cells can be the automatic repair of DNA done by the cancer cells can be reduced, which will in turn enhance the chemotherapy effectiveness and also reduce the drug resistance. So this is about the DNA repair, the basics of DNA repair, why DNA repair, and the current affairs related to this DNA repair topic. Okay. Now coming to the next topic, that is the scientists have recently come up, the ICAR scientists have recently come up with a new plant genome editing tool. The name is ISDRA2-TNBP. It is a small precise DNA cutting enzyme. See, it is also like the CRISPR-Cas9 Cas12 enzyme only. Here the enzyme name is ISDRA2-TNPP. Okay, this is a small precise DNA cutting nuclear enzyme which is used for genome editing in plants. Okay, this was discovered from a bacterium called, okay, this is the this has been discovered from a bacterium. Okay, this Deonococcus radiodurans. Okay, this is the name of the bacteria from which this small DNA cutting enzyme has been discovered, okay, by the scientist of developed by the scientist of ICAR. Why it is important? Because this DNA is very small, this DNA cutting enzyme, because we all know that Cas9 and Cas12 protein will act as a DNA cutting molecular scissors. This protein, the enzyme that has been discovered newly is also a protein, but it is much smaller than the CRISPR-Cas9 protein, so it is easier to use in the plant cells. Okay, one important thing related to this new enzyme is that it is very accurate, means it will cut the DNA at the specific targets only with a minimal mistake, whereas the genome editing techniques like using Cas9 and Cas12 enzymes, there are some errors while cutting a DNA, but this new protein, this new enzyme, that is TNPB, has a clear cut and has a specific target. Okay, efficient, it is very efficient in some of the plants like rice and Arabidopsis. Okay, and it is future ready, it means it can make the plants better crops. We can able to make better crops stronger, healthier, and more nutritious, because we can include more traits like drought resistant, high yielding varieties, climate resistant crops. So on this lines, this new enzyme, DNA cutting enzyme TNBP, becomes very important from your exam point of view. Okay, now coming to the next topic related to the organ on chip technology. See, organ on chip is nothing but a small lab made devices which will mimic the functions of human organs using a living cell. Human organs, for example, lungs, heart, kidney, it will be replicating, or it will be mimicking the functions of this human organs. Okay, why this becomes very important? Because see, if a device that can mimics the functions of human organs, the scientists can study the device efficiently without using any animals for the development of new drugs. In this concept, this organ on chip technology becomes important. Okay, what are some of the specifications of this particular organ on chip technology? It focuses on accurate drug testing, because it can test how drugs can affect the human organs, providing more reliable results. Why? Because it mimics the original functions of the human organs. Okay, it also helps in disease research. How? Model diseases, we can create a model diseases for cancer and diabetes, and that can be checked in this particular device which is mimicking the original human organs, which can lead to better treatments. It can also provide personalized medicine. How? Because the organ can be, because we have seen that it has been made from using live cells. For example, if a patient is suffering from some kind of cancer, the living cells from that a patient can be collected and an organ can be developed mimicking that particular living cells. And when a living cell is made from a particular person, this can lead to a personalized medicine, because one size doesn't fit all applies to this cancer therapy also. So this helps in personalized medicine, and it is faster and cheaper. And ethical aspect, we could see that we are not going to involve any animals for drug testing, so it becomes more ethical also. So this is about this organ on chip technology. I hope the concept and the application of this organ on chip technology is clear to you now. Coming to the next topic, that is unified genomic chip. See, it is nothing but it is a single nucleotide polymorphism. It is a chip. Okay, it is a chip that is used for analyzing a genetic variation in livestock. Okay, what are the objective of this particular unified genomic chip? It is actually designed specifically for genomic profiling and genomic evaluation of Indian cattle breeds. This is an important point. Why it becomes important? Because it aims to enhance the genetic improvement and genetic potential of diverse dairy animal productions in India. What are the benefits? Here it will help the farmers to identify the high quality cattle early, and it also enhances the dairy farming efficiency. How? By selecting the animals which will have a superior traits to this genomic evaluation: milk yield, disease resistance and growth rate. Variants, there are two variants of this chip. It is God chip for cattle and Mahish chip for buffalo. And who have developed this particular unified genomic chip? It is consortium led by the Department of Animal Husbandry and Dairying, Ministry of Animal Husbandry and Dairying. And who are the partners for this particular development of unified genomic chip, God chip and Mahish chip? It is partners include National Dairy Development Board and National Institute of Animal Biotechnology. I hope the unified genomic chip and the uses of this unified genomic chip is clear to you now. Okay, so we have seen that it inserts a chip called single nucleotide polymorphism. Okay, this is a genetic variation where a single nucleotide, either it is A, T, C or G in the DNA sequence, it will be altered. How it will be working? This S and P will be occurring at a specific points in the DNA, and this can vary between the individuals. Okay, some SNPs of this genetic variation will be having a traits like disease resistance, milk yield or growth. Okay, so these single nucleotide polymorphism in the gene will be identified using these SNP chips or genetic sequences, which in turn helps in the genetic selection for breeding and to improve the traits in the animals or plants. Okay, these SNPs are the tiny genetic changes that are made into the genomic sequence, which will help in identifying the desirable traits and improving the breeding decisions. So this is about this single nucleotide polymorphism. It is a genetic variation where a single nucleotide, that is the nitrogenous basis, either A, T, C or G in the in the DNA sequences altered. Okay, this is the background of this SNP and SNP chips. Okay, how this is applied to the genomic identifying the genomic potential of a particular cattle or particular breed. Okay, so this is about this unified genomic chip. Coming to the last topic of today's session, this is an important topic, and it is about the Wolbachia bacteria. So what is this? It is nothing but a bacteria that naturally affects some of the arthropods, okay, especially the mosquitoes. Okay, this bacteria actually transmits from one generation to another through the eggs. Okay, and this can manipulate the reproduction of the host organism. This is a very important point. Because why? Because this manipulation will only cause some of the permanent effect in the host organism. Okay, how this can be harnessed? This can be harnessed as a tool to control mosquito population and reduce the spread of diseases like dengue, Zika, chikungunya. Okay, the target of this particular Wolbachia bacteria is the Aedes aegypti mosquitoes, which cause the dengue and dengue, Zika, and chikungunya. Okay, how does this work? See, scientists are working on this project. How it will work? See, the egg of this Wolbachia bacteria will be injected. Sorry, the Wolbachia bacteria itself will be injected into the eggs of the mosquitoes. Okay, here what happens, we have already seen that these bacteria in the next generation will manipulate the reproduction of the host organism. So what happens, these larval exposures are now exposed to the water in which has infused with the Wolbachia bacteria. Now the infected male will release into the wild to mate with the females. Okay, through this mating process, this bacteria will be transferred into the Aedes aegypti, that is the female of the mosquito group. Okay, what happens when this bacteria enters into the eggs of this particular mosquito? See, here it will lead to the cytoplasmic incompatibility. What does it mean? If the affected males of this Wolbachia bacteria mate with the unaffected females, the egg will not hatch. It means it will reduce the mosquito population because there will be no more reproduction happening. Okay, this also prevents some of the viruses to inhibit. Okay, inhibition of pathogens, so that this will also decrease their ability to spread the disease. So two important points related to this Wolbachia bacteria is that the eggs will not hatch, which will lead to reduction in the reproduction, and the second is that it will prevent certain viruses from replicating in the mosquitoes, thus it will reduce the spread of disease. Okay, this is the two important points which you have to note down with this Wolbachia bacteria. So what is the important sustainability of this particular method? See, the bacteria are passed down through generations, and it is a long lasting solution for controlling mosquito population, because if the bacteria is transferred from one generation to another generation, the reproduction will not happen, which will effectively help in controlling the mosquito population. Okay, so here is the visual diagrammatic representation of this Wolbachia bacteria. See, we all know the target is Aedes aegypti mosquito. When Wolbachia bacteria is when this Aedes aegypti is infected with this Wolbachia bacteria, it will bite a dengue infected. For example, it is going to bite a dengue infected person. But we know that this Wolbachia bacteria will block the dengue virus. Okay, from transmitting it from the mosquito to the humans. So if this dengue affected bacteria is going to bite another person, the person will not get dengue, because we know that this Wolbachia bacteria has a trait of inhibition of the pathogens. That means it will not allow the Aedes aegypti mosquito to transmit the viral disease. So this is about the working of how Wolbachia bacteria will be working, and how the reproduction of mosquito will be coming down, which in turn reduces the population, which in longer term may help in eradication of mosquitoes. And second is the important point, that is it will block the disease spreading nature of a mosquito. Okay, so this is about this entire concept of Wolbachia bacteria. I hope the concept of this topic is also clear to you now. So this is all for today's video, and this is all for today's lecture. In next lecture, I will be meeting with the MCQs that are made specifically from these topics whichever I have discussed. So these MCQs discussion will become very important from your upcoming UPSC CSE Prelims examination. So don't forget to watch this video, and I will be meeting you with yet another session on the same topic in the form of MCQs. Till then, happy learning. Have a great day.